Welcome to the Explainer. Right this very second, there's a bustling microscopic city operating inside your body. It's wildly complex, totally fascinating, and it's literally the foundation of what keeps you alive. We're talking, of course, about the eukaryotic cell. To really understand how this hidden metropolis works, we aren't just skimming the surface. We're distilling the heavy-hitting biochemical mechanisms straight from a highly rigorous scientific source, Advanced Nutrition and Human Metabolism, 6th edition. We're going right to the textbook data to see exactly how these incredible cellular structures tick.
Okay, so here's our map for today's explainer. We've got 5 critical districts to explore: border control, the power plants and factories, the command center, the cellular workers, and finally, a self-destruct sequence known as apoptosis. Let's dive right in.
Before we actually breach the city limits, let's just make sure we're completely on the same page. A eukaryotic cell is the basic, living, structural, and functional unit of the human body. The name itself is actually a huge giveaway. Eu means true, and karyon means nucleus. So unlike primitive prokaryotic cells, our eukaryotic cells possess a highly defined nucleus, a true centralized command center.
Moving to Section 1, border control, officially known as the plasma membrane.
This outer boundary totally encapsulates the cell. It acts as strict border control, dictating exactly what's allowed to enter and exit. And looking at this side-by-side comparison brilliantly illustrates the sheer scale of what we're dealing with here. On one hand, we've got the macro scale of human nutrition, the actual food on your plate. But zoom all the way down and you find the micro scale of complex lipid molecules forming the very walls of our cellular city. This plasma membrane is fundamentally a sheet-like structure built from these incredible lipid bilayers.
Let's take a closer look at the specific tools this border patrol uses. The wall itself is a phospholipid bilayer with a hydrophobic or water-repelling core. That's absolutely crucial because it stops water-soluble compounds from just freely washing in and out of the cell. But look closely at the cholesterol wedged right there into the membrane. It actually acts as a regulator for fluidity and mechanical stability. And see those sugar residues sticking out on the outside, the Glycocalyx? Those act just like tiny molecular antennae, picking up vital signals from the outside world and serving as specific markers so the body knows exactly what kind of cell it's looking at.
Section 2, the power plants and cellular factories.
Now that we've made it safely past border control, we move inward to the metabolic districts. This is where the city generates its energy and manufactures essential biological goods. First up, we've got the mitochondria. This double membrane structure is the cell's primary power grid, converting the oxygen you breathe and the nutrients you eat into ATP, the ultimate energy currency of the body. But here's a totally wild twist. These cellular power plants actually carry their own unique DNA. Yeah, unlike the genetic code locked away in the nucleus, mitochondrial genes are inherited exclusively from your mother. And they code for proteins that are absolutely vital for producing all that energy.
Right nearby, we run into the city's 2 main manufacturing districts, the endoplasmic reticulum, or ER. On one side, there's the rough ER. It gets that name because it's physically studded with ribosomes, making it your primary factory for building proteins. Then, on the other side, we have the smooth ER. No ribosomes here, but it's incredibly important. The smooth ER handles lipid synthesis, it pumps calcium ions in muscle cells, and it even houses enzyme systems dedicated to the crucial job of detoxifying drugs and other compounds.
Now, once those proteins and lipids are built, they've got to be shipped, right? Enter the Golgi apparatus. This functions exactly like a highly efficient post office. It consists of stacked networks. The cis network receives the freshly synthesized cargo from the ER, processes it, and then the trans network sorts and packages those goods into specific transport vesicles for ultra-precise delivery. They'll either go to other parts of the cell or be shipped right out of the cell completely. All right, Section 3, the command center, DNA, and blueprints. It's time to breach the heavily guarded nuclear envelope and step into the most secure district of them all. The nucleus is the largest organelle in the cell, and it's where the master blueprints of your entire life reside. Let's look at how the cell actually turns those blueprints into physical structures. When the cell needs a new protein, it follows a really strict sequence. First is cell signaling. The specific need is communicated directly to the nucleus. Second is transcription. The DNA unravels just enough so its genetic code can be transcribed into a strand of messenger RNA or mRNA. Third is translation and elongation.
That mRNA leaves the nucleus, finds a ribosome, and essentially acts like a recipe card. Transfer RNAs start bringing in specific amino acids, snapping them together piece by piece, like a giant puzzle to build the final protein. And to make sure this blueprint reading goes off without any fatal errors, the cell relies on some strict architectural rules called complementary base pairing. It's simple but elegant. Adenine always pairs with thymine, or uracil if we're dealing with RNA, and guanine always pairs with cytosine. This specific letter pairing guarantees that the cell can flawlessly replicate its DNA and transcribe its recipes with a mind-blowing degree of fidelity.
But, this raises a really fascinating question. If the DNA is safely locked away inside the nucleus, how does the cell actually know when to speed up production, or how to react to sudden changes in the outside world? Who's the one carrying out these commands on the ground?
Well, that brings us to District 4, the cellular workers, proteins and enzymes.
These proteins, enzymes, are the highly specialized molecular machines constantly hustling around the cytoplasm to keep this microscopic metropolis running. A massive chunk of this workforce is made up of receptor proteins sitting right on the cell membrane. Notice how they translate outside messages into inside action in a few different ways. Some create internal chemical signals, like a hormone binds to the outside, and boom, the receptor triggers a second messenger like cyclic AMP on the inside. Others act as ion channels, where a simple voltage change forces a gate open, letting ions flood in. And some even internalize their stimuli completely, pinching off a coated piece of the membrane to swallow a molecule, like a hormone or LDL cholesterol, perfectly intact.
Then we have the enzymes, the ultimate cellular catalysts. To quote our text directly here, a substrate must fit perfectly into the specific contours of the enzyme's active site. The most common analogy used to describe this is a lock and key. These catalytic workers are just incredible. They speed up the metabolic assembly lines exponentially. But get this, they themselves are never consumed or made part of the final product.
So the crucial takeaway here is that these enzymes belong strictly inside the cell. But what happens in a medical emergency? When the cellular plasma membrane gets damaged by, say, a virus or a lack of oxygen, the cell springs a leak. These intracellular enzymes escape out into the bloodstream. Because a lot of enzymes are highly specific to certain organs, doctors can actually measure them in your blood serum. For example, if a specific isozyme of LDH is elevated in your blood, medical professionals can pinpoint exact tissue ischemia, like heart muscle damage, based purely on which tiny cellular workers leaked out of the city.
But what happens when a cell becomes way too damaged to function at all, or mutates in a way that threatens the survival of the entire human organism? How does the microscopic city know when it's time to shut down for the greater good?
That brings us to our final stop, Section 5, Apoptosis, the self-destruct sequence.
As it turns out, cellular death is actually a vital, highly organized part of living. Apoptosis is programmed cell death. Think of it as a carefully controlled internal demolition of the microscopic city. We have to sharply contrast this with pathological cell death, which is just messy and harmful. In apoptosis, the cell breaks down and its parts are neatly consumed by other cells without ever spilling its internal contents into the surrounding fluid. This clean sweep is absolutely vital. It prevents runaway tumor growth and stops the body from triggering a dangerous autoimmune disaster where it starts attacking its own tissues. And connecting this all back to our nutrition, here's a truly surprising fact, straight from our source material. The active form of vitamin D is intricately involved in both these cell death and cell survival pathways. Depending on the tissue, this incredibly common vitamin acts as a metabolic switch. It helps protect healthy cells and in some clinical settings works right alongside therapies to kill tumor cells. It's a brilliant example of how what we consume literally instructs our cells on a life or death molecular level. Which leaves us with this final, really powerful question. If our cellular cities are constantly balancing life, intense metabolism, and programmed death, how might our daily nutritional choices be tipping those microscopic scales? Literally every bite of food, every vitamin, every lipid is either building up the city walls, fueling the power plants, or altering the blueprints. Definitely something to think about the next time you eat. Thank you so much for joining me on this explainer. Keep learning, keep exploring, and I'll see you next time.